Hello everyone, welcome to this session on erythropoiesis, a process fundamental to life. Today we will trace the remarkable transformation of a stem cell into a fully functional red blood cell. This journey is not only biologically fascinating but also clinically vital because disruptions in this process explain many forms of anemia and blood disorders. So, if you have ever wondered how your body makes nearly 2.5 million red blood cells every second, this lecture has your answer. Keep your attention on the cell shapes, stages you see on the slides. They are fully characterized and carefully described to guide your understanding. Erythropoiesis is the process by which red blood cells or RBCs are produced in the body. These cells are vital for carrying oxygen from the lungs to the tissues and transporting carbon dioxide back to the lungs for exhalation. In adults, erythropoiesis occurs mainly in the red bone marrow, found in the flat bones like the sternum and pelvis. In contrast, in fetus, it primarily takes place in the liver and spleen before the bone marrow becomes fully functional. RBCs have a lifespan of around 120 days, so the body must replace them constantly. In fact, about 2.5 million RBCs are produced every second to maintain healthy circulation and oxygen delivery. Now that we have understand what erythropoiesis is, let's look at how it begins. The process starts in the red bone marrow, where we find a population of primitive cells called pluripotent hematopoietic stem cells or HSCs. These stem cells are marked by the presence of a surface protein known as CD34, which is commonly found or used to identify them in the laboratory settings. The term pluripotent means that these cells have the ability to develop into all type of blood cells, including red blood cells, white blood cells, and platelets. However, to become a red blood cell specifically, they need to receive certain signals that push them in the erythroid direction. One of the key signaling molecules is called stem cell factor, SCF. SCF binds to a receptor on the surface of the stem cell called c-KIT. These binding interactions helps direct the stem cell toward the myeloid lineage, which includes red blood cells. In addition, two other important factors, growth factors, interleukin 3 or IL-3 and granulocyte macrophage colony stimulating factor or GM-CSF support the proliferation process and survival of these early stage cells. These signals prepare the stem cells for further differentiation into red blood cell progenitor. From clinical point of view, this step is critical. If the stem cell pool is damaged, for example, due to chemotherapy, radiation, or certain viral infections, it can lead to a condition known as aplastic anemia. In this disorder, the bone marrow fails to produce enough blood cells, including red cells, which results in fatigue, infections, and bleeding. Once the hematopoietic stem cell commits to the red blood cell lineage, the next stage involves the formation of erythroid progenitor cells. These are more specialized than stem cells and are dedicated to becoming red blood cells. There are two key types of erythroid progenitors we need to understand: BFU-E and CFU-E. So first comes BFU-E, which stands for burst forming unit erythroid, E for erythroid. This is an early erythroid progenitor cell. It gets its name from the large burst-like colonies it forms when cultured in a lab. At this stage, the cell is not very sensitive to erythropoietin or EPO, the main hormone that stimulates red blood cell production. Instead, BFU-E relies more heavily on IL-3 and GM-CSF to support its growth and proliferation. As the cell matures, it progresses to the CFU-E stage or colony forming unit erythroid. This cell is further along the erythroid pathway and has high sensitivity to erythropoietin. That's because the expression of EPO receptors increases so much significantly on its surface, making it highly responsive to the EPO stimulation. This stage is very important clinically. In a condition called Polycythemia vera, a mutation in the genus kinase or JAK2 gene, which allows the CFU-E cells to multiply without needing the erythropoietin. This leads to excessive red blood cell production, which can thicken the blood and increase the risk of blood clots and stroke. So overall, the BFU-E and CFU-E stages mark the early commitment and rapid expansion of red blood cell precursors, with EPO sensitivity becoming stronger as the cells mature. After the CFU-E stage, the next key milestone in erythropoiesis is the formation of proerythroblast. Okay, so this is considered the first morphologically recognizable precursors of a red blood cell when viewed under the microscope. The proerythroblast is a relatively large cell, about 20 to 25 micrometers in diameter, making it one of the biggest cells in the red cell development pathway. Its cytoplasm stains deeply basophilic or blue due to high concentration of ribosomes, which are actively producing proteins. Inside the nucleus, the chromatin is open and lightly stained, which reflects the high transcriptional activity, and you can typically see one or two prominent nuclei, small round structure involved in making the ribosomal RNA. This stage marks the initiation of hemoglobin synthesis. The cell begins producing both alpha and beta globin chains, which will later combine with the heme to form functional hemoglobin, the oxygen carrying molecule for red blood cell. From histological point of view, the proerythroblast appears as a large round cell with a rich blue cytoplasm and a central nucleus, earning it the nickname big blue cell in the bone marrow smears. Following the proerythroblast stage, the cell undergoes a series of well-defined transformation through three main stages of erythroblast maturation: basophilic, polychromatophilic, and orthochromatic erythroblasts. Let's start with the basophilic erythroblast. At this stage, the cell still has strongly basophilic or blue staining cytoplasm due to high content of ribosomes actively producing globin chains for hemoglobin. Importantly, a key transcription factor called GATA or GATA1 becomes highly active in this stage. GATA1 promotes the expression of globin genes, ensuring the cell begins the synthesizing this protein that will form the hemoglobin. Next comes the chromatophilic erythroblast. This stage is marked by a mix of blue and pink in the cytoplasm, blue from the residual ribosomes and pink from the increasing presence of hemoglobin. The name polychromatophilic literally means many colors, reflecting this mixture. So another important feature here is the increased expression of transferrin receptors, which help the cell import iron and a critical element for the heme synthesis. Finally, we reach the orthochromatic erythroblasts. So at this point, hemoglobin has accumulated to such an extent that the cytoplasm appears predominantly pink. The nucleus becomes highly condensed and dark, a stage called pycnosis. And is soon ready to be ejected from the cell. Throughout all these stages, erythropoietin plays a protective role by preventing apoptosis or programmed cell death. It does so via a pathway involving BCL-XL, a survival protein that helps the developing erythroblast stay alive long enough to mature fully. After the orthochromatic erythroblast stage, the cell takes a major step toward the maturity. It becomes a reticulocyte. So the defining feature of this stage is nuclear extrusion, where the cell actively ejects its nucleus. This is not a passive process. It actively involves a coordinated contraction of the actin-myosin cytoskeletal network, similar to how muscles contract. Once the nucleus is expelled, the cell is no longer capable of division and is almost ready to enter circulation. The reticulocyte still contains some ribosomes and mitochondria, which give it a reticular or mesh-like appearance when stained, hence the name reticulocyte. These remaining organelles are involved in finishing the synthesis of hemoglobin and are gradually broken down over the next 1 to 2 days. Reticulocytes are released from the bone marrow into peripheral blood, where they make up about 1 to 2% of circulating red cells in a healthy individual. This percentage serves as a useful clinical indicator of bone marrow activity. Clinically, a low reticulocyte count can indicate that the bone marrow is not functioning properly. For example, infections like parvovirus B19 can suppress the reticulocyte production and leading to a drop in red blood cell formation. Now, the final stage in erythropoiesis is the formation of a mature erythrocyte or red blood cells. At this point, the cell has completed its transformation into a fully functional oxygen carrier. One of the key changes in this stage is the removal of any remaining organelles, including ribosomes, mitochondria. This is achieved through a process called autophagy, where the cell digests its own internal component to streamline its structure. Alongside this, the cell undergoes membrane remodeling, forming a highly flexible and durable structure. This remodeling is supported by a protein network underneath the cell membrane called spectrin. The spectrin network provides the erythrocyte with its characteristic biconcave shape, which serves two important functions. It maximizes the surface area for efficient gas exchange, and it allows the cell to deform and squeeze through the narrow capillaries without rupturing. The mature erythrocyte contains no nucleus, no organelles. It is essentially a membrane-bound sac of hemoglobin specialized entirely for oxygen and carbon dioxide transport. Its lifespan is around 120 days, after which it is removed from circulation by macrophages in the spleen, liver, and bone marrow. As red blood cells age, they begin to show senescence signals. The two key changes occurs: clustering of band 3 proteins on the membrane and exposure of the phosphatidylserine on the outer surface. These changes mark the cell for the clearance by the immune system. So the production of red blood cells is not constant process. It's highly or tightly regulated based on the body's oxygen need. The key regulator of this system is a hormone called erythropoietin or EPO. The primary site of erythropoietin production is the peritubular interstitial cells of the kidneys. These cells act as oxygen sensors for the body. When oxygen levels in the blood drop, a condition called hypoxia, the kidney cells respond by activating a transcription factor known as hypoxia inducible factor 2 alpha or HIF2 alpha. Under normal oxygen conditions, HIF2 alpha is quickly degraded, but when oxygen is low, HIF2 alpha becomes stabilized and moves into the nucleus of the cell, where it triggers the expression of EPO gene This leads to increased synthesis and release of EPO into the bloodstream. So EPO then travels to the bone marrow where it binds to the receptors on CFU-E cells, stimulating their survival, proliferation, and differentiation into red blood cells. In essence, EPO rescues erythroid progenitors from apoptosis and promotes their development, especially under stress conditions like anemia or blood loss. As more red blood cells are produced and oxygen levels are restored, the system uses a negative feedback loop to return to balance. The improved oxygen delivery leads to degradation of the HIF2 alpha, reducing EPO production and preventing unnecessary erythrocyte overproduction. This elegant feedback mechanism ensures that the number of red blood cells in the circulation is closely matched to the body's oxygen demands at all the time. For erythropoiesis to proceed efficiently, the body requires a steady supply of essential nutrients, particularly those involved in hemoglobin production and DNA synthesis. A deficiency in any of these components can lead to impaired red blood cell formation and anemia. Let's start with iron. Specifically in its ferrous form, Fe2+, iron is critical element for heme synthesis, which is the iron containing component of hemoglobin that binds to the oxygen. In the body, iron is transported by a protein called transferrin and stored in cells as ferritin. Without sufficient iron, red blood cells become microcytic, meaning smaller in size, and hypochromic, meaning they have less hemoglobin and appear pale color. Next, we have vitamin B12 and folate. These two vitamins are essential for DNA synthesis, which is especially important during the rapid cell developments or cell division and occur in erythropoiesis. So a deficiency in either of these leads to megaloblastic anemia, where the red blood cells are abnormally large and often fragile due to impaired nuclear maturation. Another important nutrient is vitamin B6, also known as pyridoxine. This vitamin acts as a coenzyme in heme synthesis, playing a critical role or key role in the production of hemoglobin. A lack of vitamin B6 can reduce hemoglobin levels and contribute to anemia, even if iron levels are adequate. Now, understanding erythropoiesis is not just important physiologically, it has significant clinical relevance. Many disorders arise when this tightly controlled process becomes disrupted, either due to hormonal imbalance, nutrient deficiency, or bone marrow dysfunction. Let's first look at conditions associated with high erythropoietin or EPO levels. One common cause is chronic hypoxia, a state of reduced oxygen availability. This can happen at high altitude in chronic lung disease or due to heavy smoking. In such cases, kidneys respond by increasing EPO production to stimulate more red blood cell formation and improve oxygen delivery. EPO levels can also rise abnormally in certain renal tumors or rare EPO secreting tumors, causing excessive red blood cell production. Now, on the opposite end, we have conditions with low EPO levels. The most common cause is chronic kidney disease. Since the kidneys are the main site of EPO production, damage to the kidney tissue leads to inadequate EPO release. As a result, patients often develop normocytic, normochromic anemia, where red blood cells are normal in size and hemoglobin content, but they're reduced in number. Finally, let's consider nutritional anemias, which directly impair erythropoiesis process. In iron deficiency anemia, the red blood cells become microcytic and hypochromic, meaning small and pale due to reduced hemoglobin synthesis. In contrast, vitamin B12 or folate deficiency leads to megaloblastic anemia, where the red cells are large, fragile, and poorly functional due to impaired DNA replication. So to sum it up, erythropoiesis is the finely regulated process by which red blood cells are formed, starting from the hematopoietic stem cells and progressing through well-defined stages like BFU-E, CFU-E, erythroblast, reticulocytes, and finally mature erythrocytes. It's controlled by key signals like erythropoietin and depends heavily on nutrients like iron, vitamin B12, folate, and B6. Any disruption, whether hormonal, nutritional, or genetic, can lead to clinical conditions such as anemia or polycythemia. So, if you found this lecture helpful and want more clear concept based explanation in physiology, pathology, pharmacology, this kind of subjects, make sure to give this video a thumbs up, subscribe to the channel and share with your friends. Thanks for watching. I will see you in the next lecture.